We're going to significantly shift gears in our discussion for the second half. And I think the stage was set very nicely by pointing out that climate change makes everything else worse. And we're going to focus now on some very specific applications of marine organisms, marine environments, and ways to potentially take some of the pressure off the environment while also feeding what remains a pressing need for food for human populations and fuel for a world where development and growth is very much a goal for most of the world's population. How can we achieve this? How can we come up with more food, more fuel, and a smaller impact on our planet? That sounds like a high challenge. And we have to do this, as we said, without a lot of agreement, but with a number of tremendous technologies coming from science, from bioengineering, from biotechnology, and agriculture. What we're going to hear now is that this also is going to require some very hard choices, and it's going to require a lot of research to understand what the costs and benefits and opportunities of those choices are going to be. Some of these are going to be counterintuitive. If we said a lot of what's intuitive, a lot of our fears, a lot of what we think to be true may or may not be. And then at the end of the day, we're going to have science, we're going to have scientific consensus, and we're going to have a way to make business decisions and policy decisions that we hope are based on evidence, that we hope are rational. That is really a strong order, a really hard thing to do. But that's my goal, is to have that discussion here. And really, we're going to start out with a topic. Ron Stotish, who's the CEO of AquaBounty Technologies. Ron, you don't know this, but I've been assigning your product to my classes for years as a test question. Can they figure out what you're actually doing? And it's part of our genetic engineering section, and some of the students here hopefully got those questions right. But this is something that I do want our students to learn, and I think to think about it critically, to understand its capabilities, to understand how it fits into our perceptions of risk and benefit, and ultimately into public policy. Ron has a long history at AquaBounty, formerly at Fort Dodge Animal Health. He started his career at Merck, as did our last speaker. And he's a member of the World Aquaculture Society, the Global Aquaculture Alliance, as well as the AAAS. And it's a real pleasure for me to introduce Ron Stotish.
Well, thank you for the invitation to join you today, and thank you very much for that kind introduction. And I hope you'll allow me to review the responses to some of those assignments, because maybe some of your students have had some ideas that we might like to take a look at. As Fred said, I'm going to talk to you something that is near and dear to AquaBounty. We are sort of the pioneer for what we hope will be the world's first FDA-approved genetically modified food animal. Now, having said that, the title of my talk is AquAdvantage and the Blue Revolution, and I'll talk to you a little bit about that Blue Revolution, which is perhaps more in keeping with the tone of the discussion today. And if I can remember how to use this, oops. The first thing I want to remind you, and Fred already brought it to your attention, is we're talking about food. And there is global competition between a sustainable environment and being able to feed a growing world population. That's compounded by the fact that we have developing world economies in the third world where there's an increased pressure for high-quality protein diet, getting away from the starch-based diets that have been historically the basis for diets and food for people in impoverished countries. Global salmon production, and I hope everyone here at least likes salmon, whether you like it a lot or a little bit, but global salmon production is over a $10 billion business. Combined, the world salmon farmers produce over 2.1 million metric tons of salmon a year. Now, this is Atlantic salmon, and many of you may not make the distinction, but there are two major commercial sources of salmon. One is the Pacific salmon. Those are Oncorhynchus. Those are more related to the trout. The Atlantic salmon is the species that's pictured here. That is exclusively a farm species, and we'll talk a little bit about why that is. Atlantic salmon is a major cultured fin fish. In 1982, there were 8,400 tons of wild caught. That was down to 1,300 tons in 2013. And in 2015, it's probably closer to 500 tons. There are very few wild caught fisheries. There's a small fishery off the coast of Iceland and Greenland, and a small fishery off the coast of Northern Europe. The rest of the world's wild caught fisheries, and there are recreational fisheries. People always come up to me after my talk and say, but I know I fish for salmon in the Great Lakes, or I fish salmon here or there. Those are recreational fisheries. They're not commercial fisheries. Some of those are stocked. Some of those are native populations. But the important part is man has destroyed the habitat, overfished, and otherwise destroyed the wild-caught population of salmon. And in fact, in 2000, the state of Maine and the U.S. government declared Atlantic salmon an endangered species. It represents 90% of farm salmon, and it's about 50% of the salmon that is sold commercially. The shameful statistic is the U.S. imports more than 97% of the salmon that we consume. We import more than 95% of all the seafood that we consume. And we import it from some very interesting places, as I'll show you in a moment. The point that I'd like to begin to drive to is that there is a role for aquaculture, as was mentioned by the first speaker. And there are variations in aquaculture and many ways to approach aquaculture, and it's not a one-size-fits-all business, but there are opportunities. Marine protein is a healthy source of a popular and nutritious food stuff for the growing world population. And if we're to preserve that resource at the same time that we're preserving those oceanic resources that we cherish, we have to find better ways to feed our populations. I will remind you that the capture fisheries have been flat. The slide goes back 5 years. The capture fisheries have been flat at about 90 million tons for more than 20 years. And as Dr. Hilgard said in the beginning in her talk, more than 90%, and I think the Monterey Bay Aquarium statistic is that 90% of the world's fisheries are either at capacity, overfished, or stressed and at risk of collapse. That's not a very encouraging statistic. The New England cod fisheries is a very good example of a collapsed fishery and a wonderful resource that's been lost. The aquaculture, on the other hand, has been growing at roughly 7% a year for the past 20 years or so. About 90% of that seafood that's produced, both by wild caught and aquaculture, is for human consumption. About 10% for non-food uses, some of it for fish feed. And the per capita fish or seafood consumption is about 19 kilos a year. Interestingly enough, in the United States, it's about half that, which is also something that continues to amaze me. We have a growing population. We're between 7 and 8 billion people now. By 2040, according to the Census Bureau, we'll be over 9 billion people on the planet. We've already agreed that oceanic resources are stressed, terrestrial resources are stressed. There are demands for improved nutrition and improved protein in the diets of emerging countries and emerging middle classes. How are we going to feed that additional 20% or 2 billion people? It presents a problem that there are many approaches to, of which one is the use of modern molecular biology and molecular genetics. It's not the only solution, it's part of the approach that we can use to try to meet those global food security needs. The blue revolution is really the gap. If we hold per capita seafood consumption at that 19 kilos per year, the current aquaculture production and maximal sustainable capture fisheries will only account for about 50% of that need by 2040. We have to basically double production and double total seafood production. And if the sustainable fisheries are already at risk, the only way to do that, in my view, is to the sensible application of aquaculture in various forms. That is, in fact, the blue revolution. Where is aquaculture practiced? Historically, the Far East has been not only an early adapter, but for hundreds of years it's been a popular activity, for instance, in China. And 60% of the world's aquaculture occurs in China. About 90% of that number in inland aquaculture and about 10% in mariculture or sea cages, as you saw in one of the earlier presentations. An appalling statistic here is that the United States only accounts for 0.6% of the world's global aquaculture. We have the resources, we have the technology, we do not have the will, we do not have the awareness. Some of what I'm about to tell you this afternoon, I hope, will increase the ability to make intelligent decisions and policies in that area. Fred made a very interesting point in the beginning of this discussion. Science is only a part. Acknowledgement is only a part. Intelligent policies must be developed if we are to meet the challenges of natural resource preservation, sustainability, and global food security. And science and good science can't just be a soundbite or a buzzword when we're advocating for a particular partisan political position. We have to do good science and we have to mean it, and it has to be reflected on our public policies. I need to tell you a little bit about the salmon life cycle, just to kind of kick off why our project was undertaken and why it has value. Salmon start their life, obviously, as fertilized eggs. Those eggs hatch at about 30 days, yolk sac fry. They absorb their yolk sac. In the wild, they would spend about a year or two in the gravel, hiding from predators, only eating when the weather is warm, the water temperature is warm, and the food conditions are favorable. So whether it's a Pacific salmon or an Atlantic salmon, the growth is dependent on photoperiod, water temperature, and availability of food. Not surprising. Even when you put them in a hatchery environment, these fish grow very slowly in the first 2 years of life. It's typical to see a 2-year-old fish at 200 grams or so. In the wild, again, in nature, the fish would go out to sea at about 200 grams. They would undergo a process called smoltification, where they adapt their physiological processes to growth in seawater. They go out to sea, they thrive in the oceanic environment, grow to maturity, and as adults, they return to the place where they were spawned in a remarkable sort of continuous life cycle. As I've said, there are wild salmon, but if you're eating a wild salmon, a wild Atlantic salmon, you caught it yourself or someone else caught it for you. 97% of the Atlantic salmon that's consumed are produced in aquaculture facilities. Where are those facilities? You've already seen one picture of sea cages. I'll show you one more. This is in the fjords of Norway, which is an absolutely beautiful place. Sea cages, tenders. There are wellboats and service vessels that service these. The fish are put into these anchored and suspended cages. In Chile, you see much the same. You see the tender, you see the cages in the coastal areas. You have the same support network and so forth. Now, this is sort of a cafeteria for seals, sharks, and other predator species. So they, in fact, do get into those cages. They do rip the cages open. Salmon get out. In the case of Chile, Atlantic salmon are not native species. So you're introducing perhaps 100,000 escapees into an environment. So far it doesn't seem like they've established viable populations or sustainable populations, but escapes are one issue. Other issues associated with this sort of aquaculture, and it can be done well, and in many places of the world it is done well, but the biosecurity issues are critical because you have virus diseases, bacterial diseases, fungal diseases, little tiny copepods, crustaceans that are inappropriately named sea lice, which can be transmitted from wild fish to farm fish and so forth. They kill young fish in particular because they basically are parasitic organisms and sap the vitality. So there is an opportunity to practice viable aquaculture, but it comes at a certain cost and a certain risk as well. So these are some of the concerns, and I've mentioned most of them. The contemporary industry is focused almost entirely on that sea cage. It's perceived as a low input system because, after all, you put a net in an ocean, the tidal motion of the water moves stuff out, but the unconsumed feed, medication ends up in the basin below the cages. You have the possibility for disease transmission, and you have the biological diversity issues that I mentioned. So headlines from various papers around the world, you see examples of sea lice, outbreaks of ISAB. ISAB has wiped out every major salmon producing part of the world at one time or another, wipes out an entire geographic area. So these are serious issues associated with contemporary aquaculture. Where are the fish grown? Atlantic salmon are grown in Chile, in the south of Chile. That's a cold water fish. They're grown off the Maritimes in Canada. They're grown off the coast of Norway in the fjords, the UK, the Faroe Islands. There are some Atlantic salmon farmed off the west coast of Canada and BC. They're not very popular there because the Alaskans don't like aquaculture, and there are many issues of alleged disease transfer between wild and captive stocks. So this also means, as you're growing these cold water fish in these supposedly low input operations, in order to get them to viable markets, you are literally loading up 747s in Portamont, Chile, flying those to New York, flying them to Europe, flying from Oslo to New York and to other US markets, and flying also to the Far East. This is sort of the hidden cost associated with that, because the cost of transportation is enormous. It adds at least $1 or $2 per kilo to the cost of that salmon. More importantly, the carbon footprint associated with flying 747 a day out of Portamont, Chile to North America is enormous. So when you factor all of those things in, this is not the ideal environmental sustainable solution to the production of Atlantic salmon. The transition here is that many years ago at St. John's University, the Memorial University in St. John's, Newfoundland, there was a group working on some antifreeze proteins in Arctic fish. They were very interested in the properties of these antifreeze proteins. And someone got the idea of, what about a faster growing salmon? It was just sort of an adjunct to that project. It was Garth Fletcher, Choi Yu, and Peter Davies. They used some of the regulatory elements from some of the genes from the antifreeze proteins that they were looking at in order to drive expression of a salmon growth hormone gene. And they did the following experiment. They produced a construct which contained the Chinook salmon growth hormone gene. This is a salmon gene now going into a salmon. This is a promoter from an antifreeze gene and the terminator sequence from that antifreeze gene. So this 4 kb, 4,000 base pair construct was micro-injected into fertilized salmon eggs, and then they looked for fish that took up DNA and had the appropriate phenotype. And then they looked at those fish to see who had incorporated it into the germline, where it was heritable. They identified a founder fish, and from that founder, we've been breeding that fish for more than 25 years in our hatchery in Prince Edward Island. So the molecular biology and the transgenesis was here. The conventional breeding started there. I'll talk a little bit now about characterizing this fish. This is a picture, and we were joking before this. This is either a really great picture or a really bad picture, depending on whether you're for or against. And these fish are sisters. They're from the same spawn. 2 heterozygous salmon were crossed. The fish in the foreground does not carry the AquAdvantage gene construct. The fish in the background does. The difference in mass at 1 year, this is a 300-gram animal. This is over 1 kilo. The feed efficiency of the animal in the background is 20% greater than the animal in the foreground. So this fish not only grows faster, it grows more efficiently. It uses 20% less food to get to the same weight. When they did the original experiment, they were only looking for faster growing fish. What we realized after I joined the company and we started to work on the formal development of this is that this rapid growth rate essentially facilitated growth in land-based contained aquaculture systems. This was a way to beat the programming of the native salmon in that slow growth period in its first 2 years. The bonus was the improved efficiency in the utilization of nutrients. We breed the fish, maintaining the broodstock and PE in the same way that every other salmon hatchery breeds fish. So we'll show a little sex here. The eggs are collected from the females, just as they're collected in hatcheries around the world. The milk is collected from, and I will tell you, this is a homozygous neomale. It's not a male fish. It's genetically female, but it produces sperm. And I'll tell you a little bit about that in a moment. We do in vitro fertilization, mixing the eggs and the milk, and the eggs are fertilized. At a defined point after fertilization, we take those eggs and put them in a chamber, and we expose them to a hydrostatic pressure. When we do that, it effectively causes the fish to become triploid. Triploid fish produce non-viable progeny. They can't breed. They're functionally sterile. We have validated this process to greater than 99%. The eggs are then incubated normally, and this is the final product. This is a fertilized egg that produces a fish, and I'll tell you a little bit about that fish. It's all done in our PE hatchery. It's a 10,000 square foot hatchery in Prince Edward Island, which is a very beautiful place. And if you ever have the opportunity, you're certainly welcome to come visit. Also there we have a laboratory where we do all the molecular biology, we do the method development, the assays, and all of the assays that are necessary to support this product development and application, as well as our basic research for future products. A topic for another day perhaps. Now I'll come back to the point I made a moment ago. The AquAdvantage broodstock starts with a homozygous neomale. This is a female that carries 2 copies of the gene. Very early in her life, we treat her with testosterone for a very short period of time, and she undergoes what's called sex reversion. She's genetically female, but she produces sperm when she matures sexually. So we use that sperm. You now have the sort of the incongruous combination of female sperm, XX sperm, that you can use to fertilize the eggs from normal wild-type salmon and produce that AquAdvantage fish. That, after the triploidy process, we actually assay every batch of eggs to show that it's greater than 95% triploid. We typically hit greater than 99% triploid. And after those assays are completed, those eggs would be then available for release for eventual grow out. So that's the overall process. The definition of the product then is a triploid, hemizygous, all-female salmon, single copy of the construct. And I should remind you, this is 4 kb out of a huge genome. That insert represents less than 1/10,000th of 1% of the total salmon genome. So it's not a big deal in terms of the total genetic compound. And we've shown the durability now over 12 generations. The gene's in the same place, it has the same function, and the rapid growth phenotype is consistent. The conditions for use, I'm sorry, got ahead of myself. The conditions for use is that these will be grown in land-based contained systems. Normal salmon grow too slowly to be grown in recirculating aquaculture systems. If you're in a recirculating aquaculture system, you're using electricity, you're using pumps, you're using a lot of feed, you're using a lot of other components. Basically, the economics of the process preclude growth for a 3 or 4 year grow out period. If your fish grows faster, as ours does, you suddenly jump over those economic barriers and you can grow this fish in recirculating aquaculture facilities. A faster growth rate, shorter time to harvest. You have an improved feed conversion, that gives you a better cost position and a lower cost of production. You have sterile monosex populations in contained facilities. Why did we do that? We did that because we knew that the environmentalists and all of the people who oppose this sorts of technology would run around with their hair on fire if they thought that we would release fertile animals into the environment. So this is a unique animal that we could mitigate all of the environmental risks and grow this in a sustained aquaculture system, tanks if you will, and hopefully be able to address the needs of aquaculture. This enables that economic land-based cultivation. Most importantly, it reduces that transportation system that you have to have from shipping salmon from one continent to another. This is a typical RAS system. These are huge tanks. You have walkways over the tanks. Most of these are computer controlled. This technology has been evolving. Fortunately, it's been evolving as our technology has been maturing. So the timing of the engineering for the recirculating aquaculture systems has been concurrent with the development of the AquAdvantage salmon. If you look at it from an aerial shot, it looks something like this. It looks like a modern plant. You have the aquaculture tanks in here, you have a hatchery here, you have a storage facility here, you have your feed storage here, and you have waste storage there. It could have a few acre footprint, and you can grow 1,000, 5,000, or 10,000 metric tons per year in a land-based contained system. There is one trade-off. You have to be in a location where you have available water. Even though the recirculating systems recirculate 95 to 98% of the water, you need to have a source of high-quality water. You also need a low utility cost. We've identified places, for instance, in North America where you have utility costs that are 5 cents a kilowatt hour. That means that our cost of production in a system like this is actually lower than those supposedly low-cost, low-input sea cage systems, even before you get to transportation. Now, the other bonus is that you can put these facilities closer to population centers. So you could grow AquAdvantage salmon outside Boston, outside Chicago, outside pick a city, Minneapolis, Pittsburgh, and you could grow it with a small environmental footprint. you could reduce the transportation cost enormously, and you can essentially eliminate that huge carbon footprint of flying these fish all around the world. This is the topic of an FDA application. That's a whole other talk about the emerging policy, the regulatory environment, and so forth. This has been a long battle. It started long before I joined the company, and I was involved in the discussion before I joined the company. But in 1995, we established an INAD. In 2010, there was a public meeting where the FDA basically said the product is efficacious, the product is safe, the product is identical to the traditional food, and the product is not an environmental risk. We're still waiting for an approval. One thing I should tell you is that this fish has been in the test kitchen of every major fish purveyor in the U.S. And I don't talk about who has tested it because they wouldn't like that. There would be activists at their door. But when it's tested in the test kitchens with these chefs in blind taste test panels, it's tested very well, as you might expect. This is fresh fish, closer to populations with a smaller carbon footprint. So, the next to the last slide basically summarizes what I just said. The environmental benefits, improved economics, facilitates land-based salmon farming. There are still parts of the world where sea cage farming will be indicated. There are other parts of the world where you'll be able to do land-based and they'll have an enormous benefit and a cost benefit. So it's not a one-size-fits-all. There will be a reduced impact on coastal areas. For instance, Norway is no longer issuing leases to coastal areas for sea cages. They're restricting that because of coastal pollution and other issues. There's more food production per unit, and the use of sterile fish completely eliminates any effects on biological diversity. And last but not least, you have that reduced carbon footprint associated with transportation. We at AquaBounty believe that, first of all, we have to use all the resources available to address global food security and preservation of natural resources. We also believe that the intelligent use of technology is an important tool in addressing those needs. And with that, I'll stop. Thank you very much.